Good morning, children. Now we'll revise the second part of the second chapter that is sexual reproduction in flowering plants. So first of all, we'll study the pistil, which are female reproductive structures which bear the microsporangium, the ovule, and the embryo sac. The female reproductive part of the gynoecium is a pistil or carpel. If there is only one carpel, it is called the monocarpellary. If there are the two carpels are present, it is bicarpellary, three carpels, tricarpellary, and so on, which we have studied in the class 11th. If the carpels are fused, it is called the syncarpous ovary. And if the carpels are free, then you might have studied in class 11th, it is called the apocarpous ovary. The parts of the pistil are the stigma, style, and ovary. The ovarian cavity is called the locule, in which the ovules are present. Inner wall of the ovary contains a parenchymatous mass of the cells to which the ovules are present, and this parenchymatous mass of the cells is called the placenta. If we look at the microsporangium ovule or megasporangium, they are the various parts of the ovule. One is a funicle or stalk by which it is attached to the placenta. Hilum is a point of attachment of ovule to the funicle. There are the integuments, one or two integuments are present which protect the ovule. As opening is present, and this is called micropyle, and this micropyle is present in between the two integuments. When the ovule turns into the seeds, a small opening is left out of that of micropyle, which help in the exchange of gases in the conditions of seeds. The chalaza is the opposite to the micropyle, where the nucellus is a mass present inside the integuments. One of the cell of the nucellus develops into the embryo sac. The embryo sac is a female gametophyte. It's located inside the nucleus. If there is only one integument, the ovule is known as unitegmic ovule. For example, in the case of the gymnosperms and compositae family of angiosperm, if there is a thin nucellus, then the ovule is known as a tenucellate ovule. And if the nucellus is massive, large in sized, then it is a crassinuate ovule. Let's discuss about the structure of the pistil or the carpel. Carpel has a three parts: stigma, style, and ovary. It is attached to the thalamus. Based on the thalamus, so this is the structure of the syncarpous ovary, where the various carpels are fused to form the. So there is a stigma of different carpels, and this is the apocarpous condition, where each carpel is different. Each carpel has its own stigma. This is anatropous ovule. So here it is the funicle or the stalk. Ovule is attached to the funicle at a point called the hilum. This is the hilum. The outermost covering are called the integuments, outer integuments, and the inner integuments on the both sides. There's the outer integuments and inner integuments. There is space called the opening, called the micropyle. These two integuments enclose us mass of the cells, which are known as a nucellus. Now you should remember the nucellus, cells of the nucellus are diploid. All the cells of the nucellus are diploid, and one of the cell of nucellus will undergo to meiosis to form the embryo sac. So embryo sac is a haploid structure. So here is the micropylar end or micropylar pole, and the opposite to is the chelazal pole. Now, depending on the orientation of the ovule, we have the four types of the ovules. One is called the orthotropous ovule. In the case of the orthotropous ovule, the micropyle and chelazal end and the funicle, they all remain in a straight line, while in the case of the anatropous ovule, the funicle and the micropyle forms a 180-degree angle, while in the case of the amphitropous it forms a 90-degree angle. Micropyle forms a 90-degree angle with the funicle. Compylotropous, there is a small angle between the funicle and the micropyle. The formation of the megaspore or the embryo sac is known as the megasporogenesis. This megasporogenesis is the meiosis through which the megaspore or the embryo sac is formed. The cell which undergo the meiosis is known as the megaspore mother cell or MMC. So here is a sequence of the events which takes place during the megasporogenesis. So cells of the micropylar end, one cell of the micropylar end acts as a MMC. This MMC undergoes meiosis 1 and divides into two cells called the dyad. Then the second meiotic division takes place to form the megaspore tetrad. Out of these four cells, three inner degenerates and fourth act is a functional megaspore, which will again divide by the mitosis to form the embryo sac. The functional megaspore undergoes meiosis and mitosis, and this mitosis is a free nuclear type. That is, after each cell division, the cell wall formation does not take place. So nuclei, they remain floating in the cytoplasm. Hence it is called a free nuclear cell division. Two nuclei. So after first division 2 nuclei formed, after second division 4 nuclei formed, and after next division 8 nuclei formed, and later the cell wall formation takes place, and two central nuclei, they come to the center and fuse to form the central cell. The three cells near the micropylar end forms the egg apparatus, that is one egg cell and the two synergid cells, while the three near the chelazal end form the antiporals. So at maturity, there are in the embryo sac there are the 7 cells are present, but it is 8 nuclei. L is missing. Okay, so here is a nucellus. The one cell act as a megaspore mother cell. This megaspore mother cell will divide to form the two cells called the megaspore dyad, then the 4 cells megaspore tetrad. Out of the 4, 3 degenerates and only one acts as a functional megaspore. This functional megaspore divides into The nucleus of this divided into 2 nuclei, then 4 nuclei by mitosis only, and the 8 nucleus. Out of the 8, 2 get arranged as a center to form the polar nuclei, and around this the cell wall formation takes place, which is known as the central cell. 3 nuclei at the micropolar end form the egg apparatus, or with the two synergids and 1 egg cell, and opposite to the micropolar end, that is at chalazial end, the 3 nuclei form the 3 cells called the antipodal. So this is the mature embryo sac. Now you should remember that the whole embryo sac is a haploid structure. All the cells inside the embryo sac are the haploid. These are formed by the meiosis only. So at the chalazial end, there the antipodals are present. All antipodals are haploid, having the n number of chromosomes. The largest cell is the central cell, having the 2 nuclei called the polar nuclei. At the micropolar end, there is the egg apparatus having the 2 synergids and 1 egg cell. So 3 cells are present here. So total 8 cells are present, 7 cells are present, and 2 nuclei are present at maturity. There is one called the filiform apparatus, which help during the fertilization or entry of the pollen tube into the egg apparatus. So that is all for today. Thank you.